to that as well. Okay, so for now, I'm gonna do about 15-20 minutes of lecture, and then we'll take a break afterward because it's kind of early on in the class right now. So let's go ahead and start talking about your blood. So we're kind of shifting gears toward the cardiovascular system, but for us to understand cardiovascular system, we also need to understand what flows inside of it, which is our blood. So we will look at some of the characteristics of the blood, what happens to the blood, how do we form it, how do we break it down, and then what are some of the major blood vessels in our body. So what are the functions of your blood? It is important in terms of transporting gases like oxygen, carbon dioxide, your hormones, nutrients, things like glucose, fats, triglyceride, cholesterol. It removes your metabolic waste, things like urea, uric acid. So transport is kind of already in there. I think a lot of people are already comfortable with that. A few other ones that might not be as I guess easily come to mind are regulation of pH. So pH is basically your acidity or basicity of the blood. First of all, do you guys remember if your blood is acidic or basic? Anybody remember the number for the blood pH? It's basic. Anybody remember the number? It is basic, but what's the number? 7.35 to 7.45, but yes, you're close enough. So yes, your blood is a slightly basic, and for you to keep that basic blood, you do refer to your buffer system, if you guys remember that. The buffer system for your blood is bicarbonate, which was actually an exam question for you guys in exam 1. So it may come to haunt you again. It also restricts your fluid loss at the site of injury. So restriction or fluid loss or preventing fluid loss at injury or at injury site has to do with your platelets. That's something else you guys already learned about. So this is basically, platelets are the component that helps you form your blood clots, right? So if you make a cut, that initial response that it basically prevents losing your blood is done by your platelets. Defense of it. Anybody remember what is the job of defense? Your white blood cells, right? WBCs. So their defense falls into your white blood cells. And then last but not least, regulation of your body temperature is also done by your blood. So we also talked about this very briefly on the first unit, but just kind of reminding you again. When you're hot, your skin is flushed, right? It looks really red. And the reason for that is you bring a lot of blood to the surface of your skin and allow the heat to literally radiate out of your face. When you're cold, what happens is you take those blood vessels and you make them really small. So you limit how much blood comes to the surface of your skin, and you don't let that heat to be lost through your skin. So you're protecting you're preventing heat loss. So regulating body temperature has a lot to do with you getting flushed when you're hot versus you kind of look pale when you are cold. So again, that's a regulation in terms of body temperature. Now, when you compare male and female, male have a slightly more blood, about 5 to 6 liters in average, compared to females for 4 to 5 liters. The main reason why the male have more blood is they have larger muscle mass. So male have larger muscle mass. How they produce the extra blood? They actually rely on testosterone. So the main hormone, this main sex hormone for male actually contributes to male producing more blood and use that blood to make sure that their muscle mass is supplied with enough oxygen. So these are again exam questions. Make sure you guys can distinguish those. So bad blood clotting versus regular blood clotting is if you have thicker blood, typically you're more susceptible to those blood clumping together inside your blood vessels and basically cause what we call an thrombosis, which basically means one of the blood vessels are clogged and that part of the body will not receive any oxygen. Versus when you're talking about a normal blood clotting, it usually happens on the site of injury temporarily to prevent from the blood vessels leaking blood either to the outside of the body or allow the person to internally bleed until the problem is fixed. So these platelets, when we talk about normal clotting, we're talking about it happens when your body needs it to happen, versus when we're talking about bad blood clots, it happens randomly because your blood is too thick or you have too much platelets in your system. Hopefully that answers your question. Okay, so what are the components of your blood? I'm not going to spend a significant amount of time on this because this should be something very basic for you guys. Talked about it in tissue. You should be already very familiar with it since you have your lab exam today. Sorry, I'm just answering the question. What causes your platelet count to be very low? Different things. Actually, sometimes chemotherapy may cause it. Some disease causes it. Some toxins on the bacteria causes decrease in your platelet count. Sometimes the components that makes the thrombosis, sorry, not the thrombosis, the thrombocytes, the actual cells, are absent, so your body is unable to make them. So it depends. There's a wide reason of why your body will not make platelets. Age has a lot to do with it. Medication has a lot to do with platelets. Platelets count. My mom takes a medication that causes her platelet count to drop. It depends. It literally depends. So hopefully that answers your question. Okay, so what are the components of your blood? So you have the cellular components, which are basically your red blood cells, your white blood cells, and your platelets. And then the liquid portion of your blood, which is plasma, which is basically what allows these cells to flow inside your blood vessels. So let's take a look at this. So first of all, if I take a sample of your blood, so as it's shown in the picture, and put it in a test tube and spin that sample really rapidly through a machine called a centrifuge. So like spin the sample for like 30,000 times in a minute, okay? Really rapid. Sorry. What you're going to get is 3 separate layers of your blood. The heaviest layer is going to settle on the bottom, and the lightest of the layer is going to be on the top. So on the bottom, in average, about 45% are going to be just your red blood cells. Yeah, red blood cells are the ones that are going to carry your oxygen. You have a very thin layer in the middle called the buffy coat, and buffy coat is going to contain your white blood cells and your platelets. So what's responsible for the defense of your body and what's responsible for blood clotting? A majority of your blood in a healthy blood sample, which is about 55%, is going to be your plasma, which is again the fluid portion of your blood. It has a yellowish tinge to it, kind of look like an egg yolk. And again, it's going to be majority of your blood in a healthy, hydrated individual. Again, notice that when you're looking at the percentage of red blood cells, notice the word, guys. Percentage of red blood cells is known as hematocrit. If you're in a physiology lab, you will see this word over and over again. Hematocrit is the percentage of red blood cells. And in male, that percentage is slightly higher than female in average. So when you're looking at a blood sample and you spin it for a male, The male red blood cell count is a slightly higher, again, because they have more muscle mass, they have more blood compared to females. And that difference is also explained by the presence of testosterone. Okay. But in average, we're going to just say about 45% of a normal human blood will be red blood cells or hematocrit. Plasma looks cloudy. Sometimes it implies there is an infection, although plasma itself is a slightly cloudy anyway. If you're talking about any plasma being more cloudy than usual, that typically imply an infection. So what is found in each of these three components? What is found inside the plasma? What's its function? What's inside the Buffy code? What's its function? What's erythrocytes? What is its function? So let's just start with the top layer, which is your plasma. So plasma is, as I said, it's kind of that it has that yellowish or straw-colored sticky fluid that sits on the top. It's 90% water. Hence, if you're dehydrated, guys, there's going to be a significant decrease in the plasma, which makes your blood very thick. So staying hydrated actually helps your blood flow easier. It makes less pressure on the heart. So if you're well hydrated, your blood is thinner because it's mostly water. And as a result, it can actually flow really easily through your circulation. Now, what is the other 10% of your blood? It has ions like sodium, potassium, calcium, chloride. It has nutrients like your glucose, again, your cholesterol, your triglyceride, your proteins are just flowing in there. And it has waste product. And again, anytime you hear the word waste, we're not talking about poop, guys, in this case. These are metabolic waste like urea, uric acid, and creatinine are typical waste products seen in blood. Okay, so learn these words. They're gonna come back to us in urinary system later. But oh, sorry, it says price acid, uric acid. I just noticed that. So uric acid, urea, and creatinine are metabolic waste products that are circulating in our blood. Now, we mentioned that plasma has proteins, and there is lots and lots of different protein, guys. We're not only talking about 3 different points or 4. We're talking about hundreds and thousands of different proteins in your bloodstream. But what is most of your plasma protein made up of? They fall into these 3 categories: albumin, globulin, and fibrinogen. 3 main categories. So albumin is going to be basically 60% of your plasma protein. Globulin is the second most common one, it's about 36%. Fibrinogen is the least, but it's still important. It's about 4% of your plasma proteins. First, and the most important thing that I always ask in an exam and a student miss, where are these proteins made up of? Where are they made at? So they're not made by plasma because most of the plasma is water. So where are your albumin, globulin, and fibrinogens made at? They're all made by your liver. Liver is your main key here, guys. So another thing to keep in mind, liver is really important even in terms of your blood. Liver, liver, liver. They're all produced by your liver, which means if you have liver damage, you will have an effect on your blood and the effective production of those proteins in your blood. So what is the job of albumin? So albumin is using what we call osmotic pressure. So understand that when I have water inside my blood and my blood vessels, I want that water to stay in my blood vessels. I don't want it to leak to the tissues that surround the blood vessel. If the water leaks out of my blood vessels and into my tissue, it causes that tissue to literally swell up and causes what we call edema. Okay? So if the water, I'm going to repeat this, if the water that's inside the plasma, that 90% of water that we're talking about here, if it leaks out of your blood, it goes into the tissue, it causes the tissue to swell up, and as a result, causing a condition called edema. So when you're talking about osmotic pressure here, what is this 60% protein albumins doing is making sure the water stays inside the blood vessels and doesn't leak into the tissue. So let me type this for you guys so you guys can have that answer in your thing. So albumin is a protein that keeps the water in the blood vessels and prevents it from leaking into the tissue and causing tissue swelling, which is known again as edema. Now, if someone has liver disease, okay, albumin production goes down, right? Oops. And that causes a person to see tissue swelling because the water moves from your blood it's hard to type and talk at the same time. It moves from your blood to the tissue. So if I say somebody has liver disease, why do they see edema? Why do they see edema? Why do they see tissue swelling? Is because when liver is not doing so well, then it's not producing enough albumin, which means the blood is leaking its water into the tissue, and that causes the tissue to swell up. Hopefully that makes sense to everybody. Okay, so going back to your question, Veronica, what causes to have a lot of uric acid in your body? Uric acid is a metabolic waste as a result of breaking down proteins. So if you have a diet that is really rich in protein, so like if you're doing keto diet, typically you do see an increase in uric acid. It's the same thing that causes gouts in many people if they consume a lot of meat in their diet. The other thing that can cause an increase in uric acid as well as urea is kidney problems. So if you have kidney failure at the earliest stages, even later stages, it just gets worse. Your body cannot get rid of the uric acid, so it just circulates in your bloodstream. So hopefully that answered your question, Veronica. Is the concept of the albumin makes sense to everybody? This is a typical, guys, don't come back to me and say, oh, we never talked about this. This is a really typical exam question, guys. Either what is the function of the albumin, or why somebody that has liver disease has edema as a sign, or has edema basically. And you should be able to connect that to the function of your blood. Now, the second type of protein we have is globulin. Globulin is about 36%, again, produced by liver, very common. The only thing I need you guys to know is that they're kind of like the taxi drivers of your blood. They are transporters, they move vitamins, metals, ions, lipids. They basically move things from one place to another where those molecules cannot easily move by themselves. So usually things that are fatty and they don't like to interact with water, globulin is gonna be responsible for moving those. Fibrinogen, even though the percentage is relatively low, it's extremely important in blood clotting. So fibrinogen works closely with your platelets to cause blood clots. So if you either have low fibrinogen or low platelets, either of those scenarios can cause to bleeding disorders. Brenda, I'm not really sure what your question is. It prevents proteins or hormones from being degraded. Can you clarify your question for me, please? So typically, I'm just going to move on. I'll wait for you to answer, to type your question. Typically, an exam question is, what component of blood is involved in blood clotting? Okay, so if I say what component of blood is involved in blood clotting, what would you answer me, guys? This is really important, guys. This is like a trick question, I guess. Okay, but the word, when you're using the word blood, what are we including? Everything, right? Everything in your blood. So it's not only fibrinogen, but it's also platelets. So the answer to my questions, what component of blood, okay, what component involved in blood clotting, the actual answer would be both platelets, right, which is part of the Buffy code, and fibrinogen, okay, both of those. But if I ask what component of plasma is involved in blood clotting, then you only have one answer, and that would be fibrinogen. Look at the questions, guys. Look at the difference. I'm not trying to confuse you. I just want you guys to understand where each of these components are present. If we're talking about blood, we're talking about the whole test tube, which includes both the platelets and the fibrinogen. But if I say which component of the plasma is involved, your answer should only be fibrinogen, or fibrinogen, however you want to pronounce it. Okay, it is 9:23, so let's go ahead and take a break before I kind of go overboard with the information. Let's pause. Okay, so we finished talking about plasma, so we're gonna continue by, sorry, one second, let me just get my, okay. So we're gonna move on to our red blood cells for the next component. So for our red blood cells, we are looking at basically 45% of your blood. We already learned as part of your tissue lab that another name for red blood cells are erythrocytes. And again, typical exam question, identify what is another name for red blood cells, or what is erythrocytes, etc. In terms of erythrocytes, or your red blood cells, I'm just going to move on to the next slide because it makes it easier. They're relatively small cells, so they're about 7.5 micrometer, which is basically relatively small. If you think about the capillaries, which are these tiny, tiny blood vessels that you have, when the red blood cells are passing through them, they actually line up one at a time. So they are pretty small, so are the capillaries. And the size of these is really important in terms of being able to pass to every corner of the body, basically, to make sure that the oxygen is being accessed by all cells and tissues in your body. So in terms of, again, the number of erythrocytes for males versus females, you expect to see a larger number for males. So it's between 5.2 to 5.8 million per cubic millimeters, versus for females between 4.3 to 5.2 cells per cubic millimeters. If you are eventually going to do physiology, you are going to get a chance to do a count of your red blood cells by doing a sample of either sheep's blood or your own blood, depending on what your instructor chooses to do. Now, if you look at your red blood cell, is not that typical round cells, rather than it kind of looks like an incomplete donut where you have that kind of a dip right at the center. It's called biconcave. So it basically means that it has these two dips at the center. It doesn't go completely through, but the importance of this is actually that having that dip at the center gives it about 30% more surface area. And this extra surface area of your red blood cells help with effective gas exchange. So if you want to basically effectively bring in oxygen and collect the carbon dioxide, as we'll talk about in just a minute, having that extra surface area is like having multiple gates versus one or two gates. So if you have more gates, it's easier for things to get in and out of your cell versus having a smaller surface area where you have less gas exchange taking place. Now, another characteristic of your red blood cell, which is again typical written question in your exam, is your red blood cells compared to some other cells in your body, they live relatively a short life. So RBC only lives about 100 to 120 days, which is, again, the time is relatively small compared or short compared to when you look at, you have heart cells that are basically from the moment of your birth to your death, or your neurons, right? The main reason why red blood cells only live about 100 to 120 days is they lack nucleus, which is basically where you have your genetic information, and organelles. And again, organelles, we're talking about majority of the organelles like ribosomes, mitochondria, your endoplasmic reticulum, etc. So all of these are basically being taken out of your red blood cell as the red blood cell is forming and developing. So if there is a damage to your red blood cell, then they can't fix themselves. I don't know. I'm typing to everybody. I don't know. Is it everybody doesn't see what I'm typing? Because it to my chat says, so you guys can see it or you cannot see it. Yeah, my chat says it's to everybody, so I don't know why you won't be able to see it. I copy and paste it again, but it's not on my end, so I'll do this. Hopefully this works now. I did type a question though. So extra surface. So this is the two things I've typed since we came back from the break. So one is the extra surface are on the red blood cells to help effective gas exchange, and then why is the short time. So do everybody see this now? These 2 points that I made. Hope so. Okay, awesome. So we're all on the same page. I want to make sure everybody gets what I'm typing. So 2 focus points for your red blood cells. Again, why is that a specific shape? To give it more surface area so you have more effective gas exchange. And then the second thing is you are looking at red blood cells that are living relatively short lives because we removed everything that's basically inside of the cell. And therefore that cell, if it is damaged or getting old, it cannot repair itself because it's missing all of those components. Now the question is, well, why did I take all of these things out of my red blood cell in the first place? Why am I taking my nucleus, my organelles, all of them out? The reason for it is to open up a space for a protein called hemoglobin. So I'm going to type this again. So all the organelles and nucleus is removed to open up space for protein carrying oxygen called hemoglobin. So if you're looking at your red blood cells, this is how I look at it. You take all the junk out of it and you fill it with hemoglobin. And why are we doing this? Because hemoglobin is going to carry the oxygen. So you want to basically maximize the space you have inside of your red blood cells to be able to carry out your oxygen. Now, again, here is the difference in terms of what microscope you use to look at your red blood cells. Here is a typical sample of red blood cell you see under the microscope that should, again, look very familiar to you guys based on what we learned. Here is an image of red blood cells lining up inside a capillary, which is, again, what we mentioned is like really tiny. This is a 3D vision of your red blood cells, and you can kind of see that dip right at the center of your red blood cell right there. And then again, here is the biconcave. So here is one side, here is the second. You have that dip to increase the surface area. Now, let's take a look at your red blood cells, but what is most of your red blood cells, as I mentioned here on the last point, is made up of, which is hemoglobin. So first of all, hemoglobin is a protein, okay? It is a protein that make 95% of the space inside your red blood cell. So literally it takes over your red blood cell. If you open up, to give you a number, if you open up one red blood cell contains 250 million hemoglobin protein. So one tiny red blood cells, which basically means one of these little pink dots that you see right here, guys, if I burst it open, I have 250 million hemoglobin protein inside of it. That's a huge number, right? Now, the next question is, well, what is this hemoglobin made up of? So I keep saying hemoglobin protein. It carries oxygen. What does it actually look like, and what does it do? So here is a complex looking image, and I want to just kind of simplify it for you guys first. Or I'll talk about the component and then I'll simplify it. So hemoglobin protein, those 250 million hemoglobin that I have three components. A component called globin, which is protein, the protein component. A heme component, which basically gives it the red color. And then the iron component, which is a carrier of the oxygen. So here is the complex version of that. So you have 1, and you can kind of see the folds, 2, 3, and 4 kind of randomly formed kind of structures, right? The brown structures. Those are your globin units. And notice 2 of them are darker brown, 2 of them are lighter brown. Those means that 2 of them are alpha, 2 of them are beta. At the center of each of these globins, what you see is this kind of red disc, which is kind of magnified right here. What you see in the red color is the heme component, which is the red pigment. Again, makes your red blood cells red. And then right at the center, you have this kind of a green circle, which you can see here, 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 and here. There's 4 of them, and that is identifying your iron, which is the atom of iron, which directly binds to my oxygen. What do you mean by alpha? I don't know what your question exactly is. So if you can retype your question for me, that would be great. So if I was to draw a hemoglobin, guys, which is usually shorthand to capital H, lowercase b, this is how I would draw it. Oops. Sorry, I want to use a different color. So I would draw 4 circles. So here is the 4 circles, which are going to be my either alpha or beta. If you just joined me, please mute yourself. Globin. So there are 4 of the units of 4 globins. Oops. So 4 globin. At the center of each of the globin, as I say in red, you have another structure. And these red colors are depicting what? They're depicting our heme, right? Which is the red color of my red blood cell. And at the center of each of these, I'm gonna have what? Sorry, it's hard to type. Iron, which is shorthand for Fe. So you guys can type it yourself. It's Fe. Sorry, harder to write. Iron, okay? So if you look at this, guys, we have 4 structures of each, right? 4 globins, 4 heme, 4 iron. So each of these iron, guys, each of these irons carry 1 O2 molecule. Each of the irons carry 1 iron molecule. So let me ask you a question. How many oxygen molecule 1 hemoglobin carries? How many oxygen molecule can a hemoglobin carry? Correct. So let's type the question. How many O2 molecule does a hemoglobin carry? And the answer would be 4. Oops, not $, 4. Great. I wish. Now the next question I have for you is how many, pay attention to this, O2 molecule does a red blood cell carry? How many oxygen molecule does a red blood cell carry? So I got 250, I got 1 billion, I'm assuming. A lot. I want a number. So how many, let me ask you this, guys. First, how many hemoglobin 1 tiny red blood cell carries? We talked about this. 250 million, right? And each of those 250 million are carrying 4 O2 molecules, right? So you do 250 times 4, exactly. So your answer would be what? 1 billion, correct. Not 1,000. I hope it's a little bit more than 1,000. So when I ask you guys how many O2 molecule does the red blood cell carry? Okay. So think about it this way. Each red blood cell has 250 million hemoglobin, and each of the 250 million hemoglobin carry 4 oxygen. So it's 250 million times 4, and the answer would be 1 billion. So I typed it for you guys so you have that information. So please, please, guys, I'm gonna give you a second to digest this. When I'm asking you the question, pay attention to the end of the question. Is it asking for 1 red blood cell? Red blood cell, the number is gonna be huge, it's 1 billion. 1 hemoglobin itself carries 1 hemoglobin carries 4 oxygen molecules. 1 hemoglobin carries 4. 1 red blood cells carries 1 billion oxygen molecule. So depends on the person. We looked at this in terms of millimeter square. That's the numbers I gave you guys in the previous slide. So it's between 4.3, so 4.5 to 5.2 million red blood cells for male, and sorry, for females, and slightly higher for male. And then again, the number is for male is going to be higher. So, but we don't look at it as a whole body. We look at it in millimeters square. Fe, correct. Sorry, I can't type. Fe stands for iron. So capital F, lowercase e, it stands for the symbol of iron. Correct. Any question about this, guys? Good. Okay, so this is, again, don't tell me like I'm trying to confuse you. You just need to read the question carefully, guys. Am I asking you for the red blood cell, or am I asking you for individual hemoglobins? Now, we already talked about the fact that your red blood cells are short-lived, right? We said that they are short-lived because you basically take out the nucleus and all the organelles out of the cell to open up space for your hemoglobin. Therefore, these cells cannot fix themselves, and therefore they die shortly after they are produced. So what is going to happen to those red blood cells after those 100, 120 days have passed? So because red blood cells is literally packed, 95% packed with hemoglobin, when I'm destroying a red blood cell, I'm actually destroying hemoglobin. And when I'm destroying hemoglobin, I'm destroying the heme, the globin, and the iron, right? The three components of the heme. So basically, when I ask you what happens when I destroy red blood cells, I'm asking you what is happening to the three components of the hemoglobin. So first thing, what is happening to the globin, which is the protein portion of the hemoglobin? So globin is actually broken down to amino acids, right? Because if I'm saying protein, globin is a protein. If I chop down a protein, we already know that the building blocks of a protein is amino acid. But understand this, guys, that I used food to get that protein inside of me. So I'm not just gonna dump it and poop it out, right? I don't wanna get rid of it. What I would wanna do is recycle it. So I have a protein, I break it down to its amino acid, and then what I do with those amino acids, I'm gonna use them to make a new protein. So again, the concept of globin is broken down to amino acids, and then you're reusing it to synthesize new protein. Now that new protein can be hemoglobin, so you can send it and produce hemoglobin, or it could be a number of other proteins that your body produces. Heme component, which was the red color, what makes your what makes your red blood cells red, first gets degraded through many steps to what we call bilirubin. Now the color of bilirubin is kind of like a yellowish color. And what it does is your liver is gonna pick up the bilirubin. So bilirubin eventually ends up in your liver, and it goes through your digestive system and then eventually excreted as part of your poop. So part of the color of your fecal matter comes from your body effectively breaking down heme component and removing it in fecal matters. And yes, it is like a cycle. For your globin, it is like a cycle, correct. So heme is the only component of the hemoglobin we actually get rid of, because it's a color component, we don't really care, we can easily make it. So heme becomes bilirubin, bilirubin goes to your liver, goes through your digestive system, and you eventually poop it out. You also get rid of part of that waste product in your urinary system. So what we consider partially the yellow color of the urine is also associated with removal of bilirubin. The last part that we have is iron, right? the component that actually carries out your oxygen. So what is your iron going to do? Again, I need that iron to produce new red blood cells. I don't want to get rid of it, right? So what do? It says it's stored in the liver for reuse, but here's the more important one. Iron can be reused by going to the red bone marrow and utilized to make new red blood cells. So when we're talking about recycling, iron is the most important component that needs to be recycled. Because if it's not recycled, if you're short in iron, you will not have enough hemoglobin, which means you will not have enough red blood cell, which means you're gonna get diagnosed with low red blood cell count, and that's known as anemia. And again, anemia is really common in people who do not take iron or food that are rich in iron. Like vegetarian are more susceptible to it. People who are vegan are more susceptible to it because they don't consume red meat. And people who are pregnant may become susceptible to iron deficiencies, and as a result, anemia. I don't know what question 2 of the Canvas is, so. If it says what happens to red blood cell after destruction, then that is the answer. But if it's something else, I don't know. Sorry. Now, the only thing that I forgot that, no, there are many types of anemia actually, Jessica, but regardless of anemia, the word anemia itself implies low red blood cell count. And one of the reasons you can become anemic is low or iron deficiency. And we'll talk about, I think there's a few slides that I might have it, or I will type it in the chat box for you guys to learn about them. Timmy definitely talk about sickle cell anemia. Okay. Okay, the only thing that I didn't mention, and I want to make sure this is clear, we talked about the function of the iron, which is carrying the oxygen. We talked about the function of heme, which is the red pigment component, which basically gives its color. The function of globin, which is not listed here, is to carry a small amount of CO2, which is your waste gas, right? So you consume O2 and you release CO2. So a small amount of CO2 actually binds to the globin portion of the hemoglobin and gets carried away. Here's a picture of, again, a lot of people get like, oh my God, what am I looking at? It's too confusing. It's not, the goal is not to confuse you, but let's just take a look at this. Take a red blood cell. Here's my red blood cell. It's gonna go into a macrophage, which is basically a white blood cell. See how it gets chopped into little pieces? One part of it is heme, right? Look, heme becomes bilirubin, right? Bilirubin goes to your liver, goes through the digestive system, and again, eliminates as part of fecal matter. Bilirubin can go to the liver and go to your kidney and basically become part of your urine. As I said, the color yellow from urine partially is associated with bilirubin. Iron, right? Sorry, iron is another component. Again, it gets transported to your red bone marrow and you produce new red blood cells with that. So far make sense? Hopefully. It looks like a complicated picture, but it's really simple, guys. And then last component, the globin portion. So the globin portion becomes amino acid, and again, you can use it to produce new red blood cells or just produce new protein. If you mean, you said, so it goes to liver and kidney. If you're talking about specifically the bilirubin, yes, they have two paths for getting eliminated from the body, either through the digestive system by going through the liver, or going to the liver and going through the urinary system and getting peed out, basically. Yes. Okay. One thing that I do want to talk about just for a moment is if you're a healthy individual, guys, what do you expect to see the ratio of your red blood cell production be to your red blood cell destruction? So what do you expect to see in terms of production and destruction of red blood cell in a healthy individual? 1-to-1, correct, the same, right? So in average, to just kind of give you a number, guys, in average, you're destroying about 2 million red blood cell per second. So as you're sitting here relaxing, if you're a healthy individual, you're destroying about 2 million red blood cells that basically reach the end of their life. Now, if you want to stay healthy and you want to maintain your red blood cell and don't end up with anemia, you need to be producing the same number of red blood cell per second in your red bone marrow, okay? So let me just type this because I want to make sure you have that information. To keep your red blood cell level constant, your RBC formation should be equal to RBC destruction, right? So that's 2 million, and again, 2 million per second destruction and 2 million per second production. Now, production, based on what we already learned in the skeletal system, production of red blood cell takes place in the red bone marrow, correct? Everybody remember that? Red bone marrow and the spongy bone? Sounds familiar? Now, the other thing that you need to know, for red blood cell level, red blood cell production level to stay constant, right? That 2 million red blood cells per second, you need a hormone called EPO. Okay, don't need you guys to know what it stands for, but it stands for erythropoietin if you are interested to know the name. Erythropoietin, as in red production. So it depends on a hormone called erythropoietin or EPO. EPO is produced, majority is produced by kidney. Okay, so EPO is produced by kidney. So your kidney produces it, it goes to your red bone marrow, and your red bone marrow produces your red blood cells. Here's the kicker, guys. If a person has a kidney disease, doesn't matter what it is, typically any disease that affects the function of the kidney has an effect on this. So if a person has a kidney disease, EPO level decreases, okay, which decrease the RBC production, right? And person become anemic, correct? So if your EPO drops, your red blood cell production goes down, which means you become anemic. Solution, how do you fix this? How do you fix somebody that has kidney disease and anemic? They actually get EPO injection. So they get the synthetic version of the hormone EPO that causes the same job as what your body would normally produce. It's pretty darn cool, right? We can inject hormones into our body to help us with red blood cell production. Can I answer any questions for anybody? Is this making sense to you guys? Clarification on any concept? Are we doing on time? Okay, okay. Okay, so guys, Please, please, even though these are not in your PowerPoints, if I'm typing, if I'm actually spending time typing in the chat box, these are questions you need to know for the exam. So if I say, why is someone with the kidney disease can be anemic, you can immediately connect that to kidney disease means low EPO, which means low RBC production, which means the person is anemic. You have to be able to make those connections now. Okay, so let's move on to our last category or last layer of your blood, which is your buffy coat, right? So buffy coat is actually made up of both white blood cells as well as your platelets. Now, there are 5 different types of white blood cells that are listed here. There are neutrophils, eosinophils, basophils, lymphocytes, and monocytes. So I want you guys to know some characteristics that help us distinguish these white blood cells from one another, either by how they look or based on what they do. So what's their function? So first of all, what you notice is when you're looking at these white blood cells, you notice that they're categorized as either granulocytes or agranulocytes. Simply put, is when you look at a white blood cell and the cytoplasm, the spaces that is inside the cell, if it has this little tiny dots inside of it, it's granulocytes. If it's look pretty clean and uniform with no dots or no spots throughout it, it's called agranulocyte, as in they're missing those granules. So granulocytes are your neutrophils, eosinophils, and basophils. And the one that are missing those little dots are either lymphocytes or monocytes. So here is kind of visualizing, helping you see that. So you have neutrophils, eosinophils, and basophils, and you can kind of see lots of dots in these images. The 2 on the bottom is supposed to be the ones that are cytoplasm is very clear. This is not the best image, but these are not actually dots. It's just the coloring, it's the staining sample, but this is supposed to be pretty clear. So that's your monocyte, which is agranulocyte. So what do you guys actually need to know for each of your white blood cells? Very brief, guys. We're not gonna go into details about them. I know it's a lot of information here, but I just want you guys to know some key facts about them. So let me kind of highlight what I do want you guys to know and what I don't want you to know. So first of all, neutrophils are going to be the most common one, and I'll go over that in just a minute in terms of percentage with you. So don't need to know that. Their job is to phagocytose or destroy bacteria, which basically means they're gonna eat the bacteria. The other thing that you really need to know about them is, I can ignore this one. So this is out. This is out. You don't need to know that. You don't need to know this. So I'm just gonna drawing a line on it. One of the main characteristics to distinguish this is their nucleus. So their nucleus can have between 2 to 6 lobes. So if you look at this purple structure that's inside, so this is your nucleus. So see, like it has 1, 2, 3, 4, 5, and 6 lobes. You can have as limited as only 2 lobes or as much as 6 lobes. That's a characteristic. So you do need to know this. And the other thing that you need to know is that these granules typically stain a light pink, which is what you see in the background. So you do need to know this. So in terms of neutrophils, the most important thing you need to know is they are the most numerous. They are going to phagocytose or eat your bacteria. They have between 2 to 6 lobes. And their granules typically stain a light pink color. You don't need to know the information about the pus. You go into more detail about them in your physiology class. Eosinophils. Eosinophils is the next type we have. Eosinophils are one of the least common ones that we have. What do you need to know? Very important, guys. These 2 points are extremely important. Actually, these 3 points are all for eosinophils important. If you have an allergic reaction, so let's say you get hives or you break out or you get a rash, one of the main things that actually stops that allergic reaction is the activity of your eosinophils. So they degrade what we call as histones that actually trigger that allergic reaction. So ending the allergic reaction is done by your eosinophils. So end eosinophils, can't think about that. The other main job that they have is they destroy or attack parasites in your body. So we're talking about parasites that are typically found in your digestive system. They actually get targeted by this. So if you have an elevated number of elevated number of eosinophils, typically, it's either a sign that you're dealing with an allergic reaction. So if you don't see any symptoms of that, that typically means the second thing, that you have some sort of parasite in your digestive tract and your body is trying to fight with that. So those are the 2 key components you need to know. In terms of a structure, the granular color of the eosinophils is red. So this is a color that helps us distinguish eosinophils from the rest of the 4 categories of the white blood cells. Basophils. Basophils are the least common type of white blood cells we have. So look at the percentage, guys, 0.5%. We simply mean if I'm actually counting 100 white blood cells, I probably won't even see a single basophil. So I have to count more than 100 to be able to see a basophil. Most important thing you need to know for this is, first, you don't need to know this, or you need to know this is the least common. You do need to know they're the one that actually trigger the immune response. So the granules secrete histamine, which is going to cause an inflammation response. So the inflammatory response, the allergic reaction that you see, the whole histone concept is coming from the basophils. So if I ask you the question, what type of white blood cell initiates the allergic reaction, then your answer would be basophils, right? What type of white blood cell ends the allergic reaction? Then your answer would be eosinophils. Hopefully that's making sense so far. In terms of the granules for the basophil, the color is purple. And you can see this is kind of completely taking over your cell. All you see is like this really purple blob rather than anything else. Now, agranulocytes, so the ones that are basically pretty clean, clear background on them. There is no granules, there's no dots or spots throughout the cytoplasm. The most important one is lymphocytes. So lymphocytes are the ones that you probably have heard of them before. They are the ones that are identified as B cells and T cells, or B lymphocytes and T lymphocytes. So the 2 main categories you have are B cells or T cells. And again, they are the most important cells in your immune system. And they are the ones that are going to fight against very specific antigens, or sorry, very specific foreign molecules, which are your antigens. So to kind of give you a hint, guys, when we are talking about getting vaccinated, like in the case of COVID, right? We're all getting vaccinated, hopefully. But in case of when we get this vaccination, the category of the white blood cells that are going to provide us protection in the long term against the things are going to be my lymphocytes. That's their job, to create what we call memory and protect us in a long term rather than the short term, what we see for the other categories of white blood cells. So again, the most important thing you need to know is lymphocytes are the most effective part of our immune system. They fight against foreign molecules looking for something called antigens. And there are two main types, B cells and T cells. And when you take an immunology course or when you review your physiology, you go into a lot more detail as far as how they do that. Monocyte, this is a really nice picture of monocyte. I like this one better than the original one. So see, the background is pretty clear. There's no granules again. The most important things in terms of characteristics of these cells is that they have a kidney-shaped nucleus, which is what you see right here, right? And what the other thing that you need to know is that they transform into what we call macrophages and go into the tissue for the purpose of phagocytosis. Sorry, that was like a big check mark. And what was phagocytosis, guys? It is kind of cute, actually. I agree. What is phagocytosis, guys? When we say phagocytic cells, what does that imply? They do kill the bacteria, but how? When you hear the word phagocytosis, what's the importance of this word? What does this look like to you guys? It's a Pac-Man, right? To me, it's a Pac-Man, right? Right? So they're literally gonna eat them. So correct. So anytime you guys hear the word phagocytosis, I think about Pac-Man. So they're literally gonna go and find the bacteria and eat them up. Engulf them, yeah. Fancy way of saying it, but yeah, they're gonna involve or eat them, correct. Is this making sense so far, guys? Any questions about anything? Any clarification? Okay. Seems like no. So I want to just kind of show you a, I know there's a few slides. I just want to, I'll go back and forth for you guys in just a second. I think my slides moved a little bit. So in terms of the frequency of your white blood cells, this is another thing that you guys do need to know is, I don't need you guys to know to memorize the percentages, guys. I don't need you guys to measure the number of white blood cells, like 4,800 to 1,100 cubic millimeter. I don't need you guys to memorize this. Other than what I already put a check mark for your white blood cells that you guys do need to know, the other piece that you need to know is the frequency from the most to the least. Not by percentage, guys, just by knowing that, for instance, neutrophils the most common and basophils are the least common. So this is the sentence we have: never let monkey eat bananas. Yeah, I know. So neutrophils, lymphocytes, monocytes, eosinophils, and basophils. I don't need you guys to tell me what the percentage is. As far as you know this order, I am fine. That's all you need to know. Don't care about the percentage goes from 45 to 72%. That's not our goal. As far as you know that neutrophils are the most common, followed by lymphocytes, monocytes, eosinophils, and basophils, that's all you need to know. Okay, so don't waste your effort and time looking and memorizing those percentages. I will never ask you that a specific or detailed question. The other thing I need you guys to know is the word diapedesis, which is written right here. This is where you basically have a damage to a tissue, and you have these white blood cells basically come out of your blood vessels or capillaries and transferring themselves to the site of the damage. So they're leaving the capillary network, the blood vessels, and going to the site of infection. So that's what diapedesis means. So these are really cool because when you think about red blood cells cannot leave the blood vessels, right? They're just stuck in there. But white blood cells have the flexibility to kind of seep through the capillaries and get out of the blood vessels and go into the site of injury, which is a really cool characteristics to have. Okay. Platelets. So there is this slide about platelets. There's a lot of information. Don't need you guys to know any of this, so you can draw a big X on it. So this is more physiology that I talk about, or when you have like significantly extra time, which we don't have. So if you're looking at our, sorry, 1 second, I just want to see this. Okay, I just keep going back and forth. When we're looking at our platelets, which is something I'm missing, I guess. Platelets, what did I say the function is? Function is blood clotting, right? And another name for it is thrombocyte. So I wrote that for you guys, so you guys want to make sure that you have that. It's already mentioned earlier in your PowerPoint. However, if you missed that, here's the information. Again, platelets are important for blood clotting, and another name for it are thrombocytes. That's the basic information I need you to know about platelets. We don't need to know anything else. Oh, and then platelets are found as part of the Buffy code. Which we already know and discussed. So just putting it in words here for you. Very good. We have about half an hour. Should be plenty of time. So blood cell formation. Blood cell formation in general, we use the word hematopoiesis. Heme imply, again, kind of coming from that whole red concept, or red, the color of the blood red. Poiesis means production or synthesis. So it's basically the production of blood cells that are forming your red bone marrow. And in a day, we're producing total number of everything together. We're producing about 1 billion new blood cells in general in one day. Just wrap your head around that, guys. 1 billion new blood cells per day. So in 24 hours, this is the number of cells we produce. Now, when we look at the origin of your blood cells, where they're coming from, they can come from two types of stem cells. One is called lymphoid stem cells, the other one's called myeloid stem cell. You are like, oh my God, another word. Simplify this for yourself, guys. Look at the word. Lymphoid should remind you of lymphocyte. So lymphoid stem cell is responsible for production of lymphocytes. Every other type of blood cells, which include red blood cells, your platelets, the other 4 types of white blood cells all come from myeloid stem cell. So look at the picture. Picture, I don't need you guys to know all this information. All you need to know, if you're highlighting it on your image, you have myeloid stem cell and you have what? Lymphoid stem cell. Lymphoid stem cell give rise to your lymphocytes, which are B cells and T cells. And myeloid stem cell give rise to your red blood cells, eosinophils, basophils, neutrophils, monocytes, which are your white blood cells, and then your platelets. This is all you need to know, guys. Don't waste your time on all the things in the middle. I'm never gonna ask you any question about this. As far as you know, what's the origin? What are they gonna possibly end at? That's all you need to know. Simplify. Okay. And then we talked about anemia, right? Low red blood cell count. What is some of the common anemia? Basically, we have what we call sickle cell anemia. I have shown you a picture. This is an inherited condition, which means it's genetically passed down from generation to generation. The most susceptible population are African Americans, and they are going to be basically what you see a normal red blood cell would be this beautiful, biconcave disc, versus when you have your red blood cells, in the case of sickle cell anemia, they have this kind of sickle shape or half moon shape. The reason for this is the defectiveness you see in the hemoglobin. So hemoglobin is damaged. I don't want you to know the detail about this. But again, sickle cell anemia, let me type this for you guys so you have that information. I don't know if I should have it on the next slide. Let's see. No. So sickle cell anemia causes the damaged red blood cell to be destroyed at a high rate because they're damaged, right? They're not normal, causing a drop in RBC numbers. So when you think about anemia, it's not the fact that the cell shape is sickled or half moons, it's the fact that these cells that look half moon, they are basically get destroyed at a higher rate. The treatment for it is basically blood transfusion. We talked about the red blood cells being low because of the kidney disease. We talked about iron deficiency, right? We talked about that one. And then we talked about just the word anemia. So I think those are the only ones that I want you guys to really focus on. Polycythemia implies that you have a significantly higher than normal or excessive number of erythrocyte. So these individuals basically have abnormally high levels of red blood cells, which makes their blood very thick. They make them more susceptible to blood clotting, heart attacks, stroke, etc. White blood cell problems, one of the common ones that you probably have all heard of is leukemia, which is a form of cancer associated with white blood cells. It could be associated with the stem cells that are producing your lymphocytes, so they're called lymphoblastic, or they're associated with the myeloid stem cells, so they're called myeloblastic. Remember when we talked about a skeletal system, I was talking about the bone marrow transplant. So this is the leukemia is basically the people that will get a red bone marrow transplant from a donor to help treat this condition. And then the other disorder that you have is thrombocytopenia, which when you have really low concentration of platelets, I think we talked about that briefly, what causes the platelet to drop, but that's abnormally low concentration of platelets. So if somebody has thrombocythemia, what are they in danger of, guys? If somebody has thrombocytopenia, what are they in danger? Bleeding out, right? So they can bleed out from a very simple cut. They can have severe bleeding internally and not notice about it until the blood pressure is too low. So either case could cause significant damage. Leukemia is typically diagnosed by just a blood test, and then they run excessive tests into the bone marrow to see what type of red blood cells damaged. Sorry, what type of blood cells damage, I guess. But anytime, it's amazing, guys. Anytime I talk about conditions, it's amazing how much information you can get from doing a simple or complete blood cell count. But what do you mean? How would they know? They look at the pathology of the cell. So anytime the person has some sort of white blood cell disorder, they typically have they become more susceptible to medical conditions in general. So when they run a blood test, they look at the number of the leukocytes and if the frequency is incorrect, or when you look at the white blood cells under the microscope, they don't appear normal, so they are pathologically. The pathology of the cell is not correct, and that's kind of a really good hint of doing that. So yeah, pathology. Almost all the cancers, when you do any type of biopsy or blood sample, it's all through pathology. Look at the tissue and see what they look like. Look at the cells and see what they look like. But yeah, like looking at it as always sick like flu, to me, it's just that generically, if somebody has some sort of disorder associated with white blood cell, your immune system significantly declines, which basically means you can't fight off any type of infection. And that's the first trigger for people to try to do a blood test. Okay, you don't need to know this. I'm going to draw an X on this one, so we can ignore this. Okay, is there any other questions? Because I'm going to switch gears from blood to blood vessels. So if there is any question about blood, let's address those now before we go to blood vessels. I don't know, yes means you have a question. Okay. Well, type away whenever you get a chance. Okay, so let's go ahead and move on to your blood vessels. So blood vessels are going to be connecting point to our cardiovascular system, which is heart. So if you have an idea about the arteries, understanding the anatomy of the heart becomes significantly easier. For anemia, if you are iron deficient, they usually don't directly go to, Jessica, are you talking about iron deficiency anemia? Okay, okay. So iron deficiency anemia, the best way to do it is, it depends on how low your level is. So if you are a vegetarian or a vegan, they usually just give you like iron supplements, which are really tough on your digestive system. They cause severe constipation. Or they just tell you to eat more green leafy vegetables. If you can eat meat, they suggest red meat, which is really high in iron. But even like when I'm pregnant, so my numbers were lower, so I'm diagnosed with like iron deficiency, but they didn't recommend for me to take additional supplements. You just tell them to change it with diet first, and then they kind of assess it. They don't immediately jump into iron pills as a supplement. But yeah, supplements is the easiest one, I guess. All right, so let's go ahead and take a look at our blood vessels. So when we look at our blood vessels, there's three main categories of blood vessels: arteries, veins, and capillaries. And I think all of you guys at some point have heard of these words. We're just gonna put them in context of what's the proper time to use each of these vocabulary. So first of all, let's start with arteries. Arteries are basically any type of blood vessel that is taking blood away from the heart. I think that's the biggest misconception I see among my students, as they say, oh, arteries are going to be red and my veins are going to be blue. For majority of the part, that is correct. There are exceptions to the rule when it comes to the heart. But typically when we do color coding on the whole body, except the heart, the red ones are going to depict our arteries, which are going to carry oxygenated blood or oxygen-rich blood away from the heart toward the body. And our veins, which are the opposite of that, are going to be the ones that are depicted in blue, and not actually blue, and they're carried deoxygenated blood, or blood that is low in oxygen, back toward the heart. But for the purpose of anatomy, if I ask you to describe an artery, Don't describe it in terms of color. Do not do that. Describe it in terms of the direction in relationship to your heart. So arteries are going to take blood away from the heart, and veins are going to bring the blood back toward the heart. So arteries, my students were saying away, right? You're moving away from the heart. And what is vein? Veins is dumping something, right? You're returning something to the thing. So away, bringing back. I don't know. If that helps for you, go for it. And in between my arteries and my veins, we have the category called capillaries, which is these tiny capillaries, the structures you see in the middle. And capillaries is the only place, this is really important, guys. They are the only place that I can actually exchange my gases, which means I can give up the oxygen to my tissue and pick up my CO2. Or I can pick up the oxygen from my lungs and drop off CO2. So that gas exchange that's happening, it doesn't happen in your arteries, it doesn't happen in your veins, it only and only happens in your capillaries. So please do remember that it's really important. Just want to make sure I don't go over the time. So when we're talking about blood vessels, there is 3 layering or tunics that are found around the blood vessels. And again, the main blood vessels we're talking about are arteries and veins here, because they're the ones that have these 3 layers very distinctly identified. Now, these 3 layers are tunica intima, and again, tunica just simply means layer. Tunica media and tunica externa. Guys, if you just look at the names, you should be able to say where they're located, right? Intima, like intimate, so they're gonna be inside, right? Tunica media as a middle, right? And then tunica externa would be the outside. So I don't need you guys to write to know this description. I don't need you to know what it is. I do need you guys to know that tunica media is responsible for vasoconstriction and vasodilation. And you don't need to know what is the externa made up of, so you can take that off. As far as you know, you have tunica intima, media, and your externa. Now this space at the center where your blood is passing through, that gap that is at the center, that circle at the center, that's referred to as the lumen. So if you're looking at a blood vessel, you basically have your lumen. My fingers right now, that is like holding the thing, that's representing the tunica intima, and the space right at the center is the lumen. Going 1 layer out would be tunica media, and then outside of that, you have your tunica externa. So if we look at the arteries, and again, arteries are the ones that are going to take blood away from the heart. We have these really large arteries, guys, that are significantly elastic. And I want you guys to understand, when we use the word elastic, okay, elastic is recoiling ability, not how much they stretch. This is a common misconception, guys. So when I say something is elastic, it doesn't mean how much I can stretch it to make it larger. It's when I release this stretching, how fast it recoils back to its original size. So when you are healthy, you have beautiful arteries, this elasticity is super high. As you age, as your blood vessels become worn out, this elasticity goes away, which means it's harder for your body to maintain blood pressure. And that's why a lot of elderly, especially anybody above the age of 80, has significantly higher blood pressure compared to a healthy adult, because they're losing that elasticity. Okay, so make sure that's clear. So the largest arteries we have in our body are called elastic arteries. And these are like your aorta, you probably have heard of it, or other major branch that comes off of your aorta. They have high elastin, which basically is a protein fiber, and it again helps with maintaining your blood pressure. That's all you need to know. So if I ask you what is the largest category of artery, you immediately have to say elastic arteries. Now here is the elastic artery comes out, and then it starts to form a smaller and smaller branches, because I can't just keep one big artery. So I need to form a smaller arteries. So as it starts to branch, as I start to form a smaller branches, I go to a layer called muscular or distributing arteries. So they start to distribute blood, as the name implies. This is commonly, basically, or I guess you say that these are the most common named arteries in our body. So when you go to the lab and you learn about the heart anatomy and blood vessels, we get introduced to a lot of terminology for blood vessels. And what you're actually learning are the name of the muscular arteries, like radial artery, ulnar artery, brachial artery, axillary arteries, etc., right? All of these are your muscular or distributing arteries. Next, if you continue going down and continue to forming a smaller and smaller branches, we get to arterioles. Arterioles literally means little artery. So you start with elastic, you went to muscular, and now arterial. The most important thing you need to know for arterial, other than the name, that the fact that they are the smallest arteries, is that they are the one that you can actually control the diameter of the arteriole by sympathetic nervous system. So what does that mean? Basically, what it means is I can constrict or dilate my blood vessel to match my pressure with my nervous system. So if a part of my body needs more blood, I can open up these arterioles and send a lot of blood through it. If I need significantly less blood supply to that tissue, I can make that blood vessel small and limit the blood supply to that part of the body. So it depends what your body actually needs. Capillaries, as I mentioned, capillaries are going to be basically the blood vessels that are essential for allowing your gas exchange to take place. So that's the only type of blood vessel where gas exchange takes place. They are tissues that are poorly vascularized. This is a common exam question, guys. Which tissues are poorly vascularized? Tendons and ligaments are poorly vascularized, which means very little blood vessels. Which tissue have no blood vessel? Avascular means no blood vessels. That's your epithelia. So your skin, for instance, have no blood vessels, the top layer, and your cartilage. So they're actually relying on diffusion to get their nutrient and oxygen. So epithelia and cartilage are really hard to heal because they don't have direct blood supply. So they cannot, basically, they don't have access to the nutrients they need to heal themselves. Time is it? Okay, well, a few minutes. I just want to see how many slides we have left. So, okay, we can finish this. This is only 3 slides. So blood-brain barrier, I'm not really going to go into it. We will discuss this in terms of nervous system later on. The most important thing I want you guys to understand about blood-brain barrier is that you do not want everything to circulate in your blood. Everything that circulates in your blood to have access to your brain tissue. Brain tissue is super important to us, right? It literally runs our body. So if you take something, you don't necessarily want that to be, your brain tissue to be exposed directly to that. So what you do is you create what they call a blood-brain barrier, a barrier that supplies your blood supply from your brain tissue. Don't need to know what they are made up of, tight junction, etc. You don't need to know that. The only part that you do really need to know for this is that the blood-brain barrier is present, but it does not, this barrier does not protect against oxygen, carbon dioxide, and some of the anesthetics. Which basically means if you have oxygen in your blood vessels, it's gonna go to the brain. If brain has carbon dioxide, that's gonna get picked up. If you're getting medication to knock you out, it's gonna go to your brain tissue and knock you out. So it is a still component that can easily go between blood and brain, but almost everything else gets blocked and then checked before it gets to your brain tissue. That's what a blood-brain barrier is. So be careful with this. If I say which component can pass through, your answer is oxygen, carbon dioxide, and anesthetics, and of course, alcohol and some drugs, unfortunately. But for most part, not everything can freely pass through this blood-brain barrier. Now, when your gas exchange has taken place, right? So whenever you think about gas exchange, again, inside your body, you're gonna give up your oxygen, right? Give it to your tissue, and you're gonna pick up the carbon dioxide, which is the waste gas. So now you need to return this bad blood, quote unquote, or deoxygenated blood back to your heart. So you're gonna use a set of networks of veins. Now, the smallest veins that picks up that deoxygenated blood are called venules, okay? So see how your capillaries are basically connected to the venules? Those are your little tiny veins that collect from the capillaries. Now, the venules are eventually gonna go to your veins and then eventually return back toward your heart. Really important thing, guys, that you need to know for your veins in general. Blood pressure in your veins are significantly lower than arteries. Think about this. Arteries are coming from the heart, right? And your heart is a pump, right? It pumps. So when you pump, what you're creating is pressure. So arteries are gonna have significant amount of pressure. Veins are coming back toward the heart, so they lost that pressure as they have circled through the body. So the blood pressure in your veins are going to be lower than your arteries, okay? So the question is, I have low pressure. So for instance, how do I take the blood that is in my toes or my feet or my leg and bring it up against the gravity back toward my heart? How do I do that when I have little or no pressure to work with? For that, this is what you use, the mechanism to deal with that low pressure. First of all, if you compare the lumen, the opening of the arteries versus vein, the veins are gonna be much larger. So they're gonna allow a lot more blood to flow in that direction. So it's kind of like you move the blood once, let's move a significant amount of it. Second thing you have is these valves right here. So see, you have one set of valves and one set of valves right here. The other thing you have is the skeletal muscle pump, which is these two muscles that you see on either side. So let me describe it to you. So let's say you have a vein in your leg, okay? And you have two muscles on either side, as you can see in the image. As you move your leg, as you walk, as you do any type of activity, these muscles are gonna contract. And what are they going to do? They're gonna squeeze the vein. So let's say they squeeze right in the middle of the vein, right? So what is that blood that is in the vein wants to do? They either wanna go up or down, right? So if you look and if the blood wants to move downward, what do you have? A set of valves, right? So blood says, well, I can't go down. So what's my other option? Go up, right? So the valve on the top opens up and the blood moves up. As soon as the blood moves out and the muscle relaxes, the blood wants to come back down again, right? It says, okay, I'm relaxed, I can come back down. What happens to this valve? It's gonna close up, which means the blood cannot come back down. So literally you have a gate and that gate's gonna close behind you. You can only move in one direction. Same thing with your arteries pump. They do the same thing as your muscles. So every time your arteries pulse because of the pressure, they're going to put pressure on the vein and move the blood upward. Same concept as the skeletal muscle pump. So kind of reviewing this couple points, guys. If I say, which of your categories of blood vessels have the highest size, highest pressure, what would you say? Which one has the highest pressure? Anybody? Arteries have the highest pressure, correct. And then arteries eventually become your arterioles, your capillaries, and eventually your veins, right? So veins are gonna have the lowest pressure. And that lowest pressure, how do I ensure that that lowest pressure can still take the blood and send it back to the heart? They're gonna use this 4 mechanism. Just one last thing to finish up with, the order of your blood vessels from the arteries all the way back toward the heart is you start with your heart, go through your elastic arteries, go to your muscular arteries, arterial, capillaries, venules, and veins. So this is the order for, and then of course heart, back toward the heart. This is the order of the blood vessels you guys do need to know from the heart and then going back toward the heart. Okay, I think we are good. Okay, that's the last slide.